Hello everyone, I am Christina Gangemi, a Science Communication Officer for the Convergence Science Network, and I'll be your host for this latest Women in Biomedicine event. I'd like to thank you for joining me from wherever you may be viewing. I'd like to begin by acknowledging the traditional owners of the lands to which we are located and pay respect to their elders past, present and emerging. So before I introduce our guests for today, I would like to encourage you to continue the conversation via Twitter. You'll see the handle and hashtag on the screen below soon. If you would like to put a question to our guests during the event, please do so via the chat facility available on the right side of your screen. So today I'm very pleased to bring you two leading researchers who are advancing the field of tissue engineering. So firstly, we have Dr. Anita Quigley. She's a tissue engineer and a cellular and molecular biologist with a special interest in neural and muscle tissue engineering. So she is currently a Vice Chancellor Senior Research Fellow in the discipline of electrical and biomedical engineering at RMIT. She's also an honorary research fellow in the Department of Medicine at the University of Melbourne and St. Vincent's Hospital, and of course part of Biofab 3D at ACMD at St. Vincent's. And we also have Dr. Serena Duchi. She's a cellular biologist with a special interest in osteochondral tissue engineering. She's a senior research fellow at the Department of Surgery at St. Vincent's, part of also Biofab 3D at ACMD, associate researcher at the ARC Centre of Excellence, at the Intelligent Polymer Research Institute at the University of Wollongong in New South Wales, and finally also an associate researcher at the National Research Council, the Institute of Organic Synthesis and Photoreactivity in Bologna, Italy. So it's lovely to have you both here today, and it'll be a great opportunity to hear more about your research and your career journey. So it was great to see your online videos detailing your research, and I'd like to first start off with a question for you both. What fascinated you about tissue engineering? Maybe we'll start off with Serena.
Yeah, so thank you, Christina, and hello, everyone. So yeah, that's a really interesting question because since I moved to Australia, I was completely new to the field of tissue engineering. I have started to work with mesenchymal stem cells back when I was in Italy, and it was all about drug delivery using stem cells and a little bit of tissue engineering using the same cells. And when I moved to Australia, in particular here in St. Vincent's Hospital in Melbourne, what brought me here was absolutely the novelty of trying to couple the 3D bioprinting technology using the stem cells, for which I'm really familiar with, while the 3D bioprinting for me was a totally new field. And so this excitement was coming to actually having the possibility to couple whatever I learned in biology regarding the stem cells and a totally new technology. And so this coupling opportunity was what excited me the most.
Oh, fantastic. Yeah, it's always great to extend on your own talents and dive into the deep end with something a little bit new, isn't it?
Yeah, sure, absolutely.
And what about for you, Anita?
So I guess similar to Serena, I started off mainly looking at cell transplantation and working with cells, particularly muscle stem cells. So we were looking at ways that we could regenerate muscle in models of muscular dystrophy in animals. So initially we started off looking at stem cell implantation and kind of quickly realized, well, after a few years, I wouldn't say so quick, but we realized we needed to move to three-dimensional implants for tissue engineering in the muscle, that stem cell injection alone just wasn't totally achieving what we wanted to achieve. And then we also moved on to nerve tissue engineering through a large grant that we got as well. So the reason we got into nerve engineering is because muscle is heavily reliant on innervation from the nerves. So we were trying to couple the two together. So I kind of moved, similar to Serena, from working on cells for cell regeneration into the tissue engineering field because, we really needed to move to not only using cells, but using scaffolds that would promote regeneration. So yeah, that's kind of why we moved into tissue engineering.
Yeah, fantastic. So Serena,
Yes.
what was it like? Because you talked a lot about the biopen and this ability that you're using at the moment and developing to be able to repair small knee defects. It's great that you've been able to channel the research, but also switch into more of a commercialization side and hopefully bringing this biopen to be used in patients in the future. So what was that experience like? I'd be interested to know, because you probably have a big background in research, but did you have any commercialization experience beforehand? What has that process been like?
Yeah, so I didn't have any at all experience in commercialization. A little bit of translation because my previous institute it is still actually an orthopedic institute where they try to translate whatever basic research discovery into something that is clinically applicable. But I had no experience whatsoever in commercialization. So for me is actually total a new field, and I'm learning. Maybe not as fast as I would like to, but yeah, we are learning all together with the team. So the good thing is that I'm not alone. So we are a team of people working on the actual biopen or Axelda project. So as you said, the main goal is to regenerate small cartilage injuries before and they become osteoarthritis in order to prevent this chronic illness. Yeah, as I said, we are a sort of multidisciplinary team. So we are biologists, engineers, material scientists, and especially orthopedic surgeons. And all together now with the commercialization pathway, we try to include in our team also special people and professional people that are more into the field of commercialization, regulatory aspects, and kit commercial manufacturer. So we are trying to, as I said, englobate many different personalities because we need to, because we are all new to the field of commercialization.
Yeah, it's very much a team effort, isn't it?
Yes, it is, and it needs to be a team effort. Otherwise, the research can't go and can't move on.
Yeah, and you said that in about 1 to 2 years, you'd like to see the biopen enter into human trials, and it's something you're actively working on. What will that trial involve? What sort of patients were you thinking to recruit? And will it be on those small bone defects in the knee, or will it be in a different
so
area?
Absolutely, this is the current vision that we have, as I said, all together with the team and the orthopedic surgeons. So we envision in a couple of years to have the first cohort of patients, and we are talking about a small group of patients, maybe like 10 or 20, to start with a phase 1 clinical trial, so to study the actual safety of the procedure. And most likely the first patients that we are gonna we are going to use as also guinea pigs. There will be people with tendon injuries, for instance, so not necessarily direct cartilage injuries, but some patients that have articular cartilage surface a little bit compromised. So we will start from these patients, as I said, to start the safety of the procedure. And then moving on, once we establish a few parameters, then we can start with the phase 2 and 3 clinical trials, so to start and measure and evaluate the efficacy of the therapy. So it's going to be, I think, a long process. So we are expecting maybe in 4 or 5 years' time to have a larger cohort of patients with small cartilage injuries.
Right, fantastic. All the best with that. I hope it goes well.
Thank you.
So I think I'll move on to Anita a little bit. You do a lot on, generating these biological scaffolds and 3D culture. You mentioned in your presentation that 3D culture is advantageous over 2D for example, a drug screening context. I'd be interested to know a little bit more on what advantage that might is for drug screening.
Yeah, well, cells behave very differently in 3 dimensions than they do in 2 dimensions. There are applications where two-dimensional tissue culture is applicable for drug screening. So, you don't have to go to 3 dimensions for everything, but certainly where you're actually looking at complex neuronal networks or interactions with cells in 3D, that can give you a very different outcome when you're using drugs, because the cells are interacting in a different way than they are in 2D. So it may be more applicable for looking at some particular drugs to do screening in 3 dimensions than in 2 dimensions. And we're particularly interested in this in the neurological field. So we're looking at screening, we're hopefully going to be moving to relatively soon, my collaborators at the Children's Hospital, some drug screening in 3 dimensions. And that's, we're all kind of working together. Again, same as Serena, it's a multidisciplinary team at optimizing these 3-dimensional cultures. So it's really, the way that cells interact and communicate with each other in 3 dimensions. It's very different to what you see in 2 dimensions.
Yeah, exactly. And I was really interested in your work with organoids, because we've seen just this huge, influx of organoid research, and you have a particular interest in studying epilepsy. You mentioned this a little bit, but maybe provide us a little bit of detail on what you're doing with these organoids for this context.
So we're working again with neurologists from the Children's Hospital as well as St Vincent's here. And with our colleagues at the Children's Hospital, we've been looking at using patient-derived stem cells. So these are cells that are derived from children with early onset epilepsy. And looking at culturing these as organoids, so we can have a look at the development of the epileptic activity in the patient cells. So these cells have been derived from skin, so they're not from brain. But using the IPS technology, we can grow brain-like tissues from these patient cells. And one of the things we're looking at is the development of the epileptic activity and how we can mitigate that in these samples using drugs. So that's in collaboration with people over at MCRI.
Oh, fantastic. So I know this may be a question for both of you, actually. How do you think we can actually engineer whole or partial organs? I know that's probably a long way off, but there is a lot of potential at the moment with what you and many others are doing. What do you think are those major challenges that could be technical challenges that might need to be overcome? I know that organs have a lot of vasculature and those sorts of things. How can we get there, do you think?
Anita, you can start.
Okay, I think you mentioned the big one. Yeah, one of the big ones is vascularization. Okay, it's keeping the cells alive in 3-dimensional scaffolds. I think second to that, innovation is very important as well. Doesn't apply to all tissues. Some tissues require more innovation. By innovation, I mean nerve supply and connection with the nervous system. Not all tissues require innovation, but things like brain and muscle, even cardiac tissue, are heavily innovated. So there's a number of challenges. It's not just as simple as growing a tissue from one cell type. We've got to get all these other cell types in there successfully as well. And I think we're moving forward, but I think that's a big challenge at the moment, as far as I'm concerned. Yeah, Serena?
Is a big challenge, even for simplest tissues like cartilage or bone, where the innervation is not, the first, let's say, feature. But anyway, so we need to reconstruct a multicellular tissue anyway. So creating the perfect environment for these multicellular systems is one of the biggest challenge, and especially to reproduce through biomaterials, whatever is considered the extracellular matrix, where the cells commonly, or in the human physiology, naturally develop and grow. So there will be a lot of research in trying to find the perfect or the optimal ECM mimicking material. So that's why we need actually to work always together with material scientists, physicists, and also mathematicians to actually model what it could be the responses of the cells to actually a new environment made with a synthetic or natural derived biomaterials.
Yeah, exactly. And Anita, I think going on to similar what you've both touched on, with these biological scaffolds and incorporating cells into the 3D printing process, how many cell types have sort of been attempted together that you may have done, or, to get this sort of complexity going?
So we've kind of going into the area of introducing vascular cells in our printing setup, but we've just started that. But there's certainly been other laboratories around the world that are a bit more advanced with this. So certainly neural cells and vascular cells have been, labs have incorporated this into the 3D bioprinted scaffolds. But one of the problems with bioprinting at the moment is the resolution. So getting them exactly where you need them to be in a three-dimensional scaffold is still a bit of a challenge. But certainly, people as well as us have looked at getting vascular cells into our 3D printed constructs. Yeah.
Fantastic. Yeah, it's amazing how you can actually incorporate the cells in the printing process and print with the cells. I didn't think that would be possible. Okay, so I might switch into some more general questions, more career-based questions. So for both of you, what sort of sparked your motivation to pursue research? Was it a particular moment? Was it a person? What was it for each of you? We'll start with Anita.
I can't say for me it was any particular moment. When I finished my science degree, before I did my honours project, I wasn't really sure about what I wanted to do. But when I started working in the laboratory, I really loved it. And during my PhD, it was very much a project where I was looking at defects in end-stage heart failure. And when I started working on things that I knew could possibly make a difference in people's lives, that really sparked my interest during my PhD. You can really generate something meaningful, something that could possibly help people. So I wouldn't say it was a sudden thing. It was more of a gradual realization that I came to through doing my PhD and working on human samples and going I used to go to where they did the heart transplant operations and get samples from people that had end-stage heart failure. And that really made me realize the potential significance of what I was doing.
Great. And for you, Serena?
Yeah, so I think it's safe for me, as Anita said, it was a gradual thing. I think that the first idea of getting into a research type of a career came after my high school degree. And originally I wanted to be an archaeologist, especially because, living and growing up in Italy, so you have ancient ruins everywhere. So it was really appealing for me to get on site and research old stuff, old things. But then eventually I got into biology. So for my bachelor, it was a 5 years degree back in the days. And during my honor thesis, I discovered the beauty of making discoveries onto living beings. And I was using at the time Drosophila, so the small fruit flies as a model system, and it was like working with alive, so in vivo models, and it was pretty exciting because I was studying development of the actual egg of the Drosophila, so it was a particularly tiny things. But then it's from there that I discovered that I was really passionate in discovering mechanism and molecular pathways involved in the determination of a final adult tissue. And I think it's during my PhD that I strongly studied how to become a researcher and how to pursue my career in academia, especially.
Yeah, great. And I also wanted to know a little bit about your thoughts on the research environment in Italy versus here in Australia. Did you notice any major differences in terms of, funding opportunities, career progression, that sort of thing?
Yeah, so certainly I did, and it's one of the reasons why for now, I decided to stay here, and I consider especially Melbourne my second home. The first one is still in Italy because all my background and yeah, my honor, bachelor and PhD, they all come from Italy. But certainly here I discovered a great country in which yeah, the career opportunities are much higher compared to Italy. Also, the competition is probably lower, considering also the amount of people present in Australia compared to Italy, and also yeah, funding opportunities are yeah. Way superior compared to yeah, Italy and Europe in general. So in Italy is changing slowly, but something, yeah, there are improvements that the governments are making and the politics and yeah, so will be possible maybe one day to come back. I still don't know. So far I prefer to stay here, but I still have I'm in collaboration back to my previous lab, the Rizzoli Orthopedic Institute in there and the CNR. So and I would love to continue to work with them because they are really nice people, and as I said, my background, it will come from there.
Yeah, we just had a question from the audience, John Neve. I think this is for Serena. Have you been made to feel welcomed in the scientific fraternity? So I'm guessing that's probably here in Australia.
I mean that if I'm yeah, I think that the environment that I found here in Australia was it was a pretty yeah, fruitful and really friendly environment as well. So, and especially the facility that I co-share with Anita as well. So the Biofab 3D or at the SMD Center is one of the yeah, the greatest environment I've been working in so far.
Yeah, and we're very lucky in Melbourne. I'd say in Australia, it's pretty much like the biotech hub of Australia. So there is quite a lot going on here and a lot of great opportunities.
Yeah, yeah, absolutely.
And let's go to the next question. What do you think is probably the biggest challenge you face as a researcher? And also, what on the flip side, what do you love most about it? What's one of the biggest advantages? So we'll go with Anita first.
Oh, biggest challenges. It's always the old nutshell of funding, but we're doing okay with funding. We've been quite fortunate. I, I, I don't know what the biggest challenge would be. I think partially being isolated in Australia, getting to collaborate with overseas colleagues is a little bit more difficult than it is if you're situated in Europe or in the States or in the UK. It's a bit of a challenge. But, you know, we're gradually overcoming that. Obviously, we're doing a lot more on the internet now, in internet meetings. One of the things I love about research is the loving coming into work pretty much every day, because, you know, there's new developments. You're always wanting to see how a particular experiment has gone or interact with your students to see how they're going. So that's really what I love about it. It's always exciting. Well, not every day, but most days it's exciting. There's some new discoveries, and that's really what keeps you going.
Yeah, exactly. Love getting that energy from the students. I've supervised students myself, and it definitely does energize you.
Yeah.
And what about you, Serena?
Yeah, so yeah, I totally share the feelings and the thoughts with Anita. And maybe on top of that, I could add that one of the challenges that I'm currently facing in the translation to commercialization and clinical translatability of our project is when you decide and when you determine that your project is finished. So basically when so the question is, when are you ready to actually move on to clinics and into patients? So the point is that there is no answer. So basically you're never ready. So you have to decide at some point what it can be applicable for a phase 1 or phase 2 clinical trial, for instance, while you keep researching in the lab. So every time there is something that is not working, either in preclinical models or in clinical human beings, so you come back into the lab and you start to understand what it is not working. So at the end, so the biggest challenge, but also what I love the most is that you never end researching. So it's a never-ending game at the end. So I think that that's the beauty of it.
Yeah, there's always another question to answer, isn't it? Never ends. But that's what it's all about. What do you think are some of actually the misconceptions about being a researcher, and what do you think the public would be excited to learn that maybe they wouldn't immediately be aware of?
Maybe the fact that there are no negative results, or there are no failures at the end. So whatever your research is about, and whatever discovery you made, whatever negative data you may achieve, or something that it goes against your hypothesis, this again is still a result, and is a positive result. So you made a progress in your research, in your scientific discovery. So I think that this might be a little bit of misconception, saying, but you couldn't sometimes, even in the easiest and the simplest experimental in vitro experiment, so you may find something that is totally the opposite of what you were expecting, but this is not necessarily mean that you have failed.
Yeah, that's really important. It's something I've had to learn too, is even if it might be not what you expected or a negative result, it's still giving you information. It's still giving you important information. So, as researchers, often there is a lot of failure before you get to the good stuff, but all those failures, they do give you sort of signposts and little hints as to where to go next. It's very important. So I wanted to ask overall of sort of where you think tissue engineering is going, and what do you think in the next, say, 10 years or so, you think we could achieve? Start with Anita.
I think certainly repair of tissues and organs is achievable in the next 10 years. I couldn't see, there have been significant breakthroughs in fabrication techniques. However, I can see definitely in the next 10 years we might be applying things like cardiac patches to repair part of the cardiac wall, or vasculature, so simple tissues rather than whole organs. I can actually see that happening pretty quickly. And in some cases, I think in some places that's kind of already happening as far as I know. I'm not completely up to date, but yeah. Which in itself, just even repairing maybe a cardiac wall or a piece of muscle with tissue engineering is a big feat in itself, and that will certainly help a lot of people.
Yeah, and what about you, Serena?
I agree with Anita. So I will talk about repair instead of regeneration, because most likely in 10 years time we will be able to repair, at least for me, for what I'm doing. So the cartilage, so the articular cartilage layer, maybe a little bit of subchondral bone for osteochondral injuries as well, but I won't still talk about complete regeneration of the adult tissue. So probably in 20 or 30 years time, I don't know if I'll still be here, but it will be interesting to see. But yeah, definitely we are talking about repair strategies.
Okay. And I wanted to actually ask you, Serena, about also your work in treating tumours in the bone. And you had this really great approach with the mesenchymal stem cells. And they basically, from my understanding, contain the cancer drugs, and then they're able to release it into the tumour. How do you engineer the MSCs to do that?
Yeah, so this was part of my project back in Italy, when we started to look to new ways to deliver chemotherapeutic drugs to osteosarcoma in particular, so which is a bone tumor that is prevalently hits young adults, so children and young adults. And we were looking to deliver the drugs directly to the bone where the actual tumor mass develops, and spare healthy tissue. And so in an attempt to also decrease the cytotoxicity given to the highest dosage that you most often you have to administer to these patients, because they when they come to the orthopedic surgeons or to the orthopedic hospitals, so is already kind of too late. So to intervene with a low regimen or low dosages chemotherapeutic drugs. So we try to tackle this delivery issue with mesenchymal stem cells, which are actually the same cells that we use for cartilage and bone repair, regeneration. And the way we actually engineer these cells is to actually provide and force them to internalize these chemotherapeutics. Like lately we have been using paclitaxel or Taxol, which is one of the cytostatic drugs mostly used in breast cancer, but not in osteosarcoma, because as I said, the dosage that you have to use in osteosarcoma is quite high. So the side effect of the drugs are quite heavily. And also we use nanoparticles, which contains molecules that are called photosensitizer, that upon light irradiation they can produce reactive oxygen species like ROS or free radicals, which are potent cell death inducers. So, and we take advantage on the fact that the mesenchymal stem cells, especially the bone marrow derived or the fat tissue derived MSCs, they can internalize quite easily these nano drugs or nanoparticles, thanks to their machinery of endocytosis, which is the mechanism that favor the internalization of molecules from the outside. And during my first postdoc, I've been studying the mechanism of endocytosis in different cells, stem cells, but also tumor cells. And this machinery, this way of internalizing in the MSCs is quite advanced. And there are different vesicles inside the cells where these nanoparticles or molecules that can be stored and still be active while the cells are moving towards the target, in our case, the tumor target, so the osteosarcoma tumor target. And another particular feature of these MSCs is that they come from the bone marrow, which is the same tissue niche of origin of the osteosarcoma tumor cells. So they kindly have an affinity, so that's why they can move towards the tumor, and once there, they can liberate, and so they can free the nanoparticles or the chemotherapeutic drugs directly into the tumor.
Yeah, that's fantastic. I mean, especially with cancer treatment, having a localized approach would be quite important, because a lot of the drugs, they have so many off-target side effects, isn't it?
Correct.
So this approach would be really important. And I'd be curious to know, because I work in optogenetics, so I'm using light I'm engineering proteins with light. I was wondering how you actually deliver the light in your experiments.
Yeah, so we have been doing preclinical animal models in mice. So we induce the osteosarcoma, the bone tumor, in the tibia of the mice. And so being such a small animal, we can deliver the light from the outside. So we are using a wavelength, which are around 700 nanometers, which are, they display the energy enough to actually penetrate the skin, the muscles, and reach the bone of the small animal. In the future, for human application, we are thinking of delivering the light through optical fibers. Mm-hmm. So there is already a technology all around optical fibers, also for many other fields and other applications. And we think that we are going to use this way of delivering the light, so intratumor, most probably.
Yeah, fantastic. We have a question again from the audience. I think it's from John Neve again, and he's asking, how important is it to look to countries other than the USA or UK for new ideas? I guess he's saying that, there's a lot of science happening in those countries, but how do we do that? Because there is a lot of research there, but there's also a lot of research going in other places.
Yeah, there's a lot of research going on overseas, and really the research community in your particular field, you generally keep tabs on new ideas that are coming up. It's very important to do this. You always want to make sure that your research is up to date and that you're not reinventing the wheel and doing something that someone has already achieved. It's very, very important to keep your research up to date. But we also have overseas collaborators that you work with, and you can bring these new ideas along together. Generally speaking, the research community is relatively friendly, and we've never really, when we've approached people overseas to do chat with them or swap cells with, even if they've got cells in their laboratory that have been recently developed, we've never had anybody really ever say no. So there's a lot of interaction with the overseas research community, and that really helps keep you up to date. It's very important.
Yeah, and I guess with science, there really is no borders. I mean, it's very easy to be able to collaborate, and whoever's doing the good research and that you want to work with, it's usually pretty easy to do that. And there are a lot of great ideas going around.
Yeah, and everybody wants to come to Australia, right? It's a great place to visit.
Yeah. We also have another question. It's actually quite interesting, and it'll be good to get your thoughts on it. Is animal testing still necessary? Why can't we just work with cells, for example, or why do we still need that?
We can't, even though we've moved to things like organoids, three-dimensional tissues, we still can't recapitulate all of the things that you would see in an animal or in the human body. So an organoid, even though you've got vascular tissue running through it, is not really connected to a blood supply. And further to that, you're missing things like the immune system. There's a whole myriad of things that you have in the body that you actually can't replicate at this stage in tissue culture. There's certain things you can, but animal research, unfortunately, is still necessary for a lot of applications.
Yeah, and you have similar thoughts, Serena?
Yeah, absolutely, and especially for surgical applications, such as, for instance, for us, the biopen application. So the passage to the preclinical animal models is fundamental. So there are several parameters that you have to understand, and that will lead to the improvement of the surgical device as well. Only when you operate a living animal. It would be nice to have more human beings, so more guinea pigs, so more clinical trials actually, and more people involved in clinical trials. So maybe it will happen in the future, but in the meantime, I mean, a passage towards the animals is necessary.
Yeah, exactly. And obviously, we don't want to have to do animal research if it's not necessary, but it has to be done with valid reasoning.
Yeah, absolutely. Fortunately, yes.
So I want to give you a little bit of a hypothetical. So say today I gave you unlimited research funding, so you didn't have to worry about the financial constraints. What would each of you do in terms of your research? What would you like to do? We'll start with Anita.
Oh, if I had unlimited funding, I'd definitely start my own laboratory, or maybe build a giant research institute. Incidentally, there is a big research institute that's going to be happening in the near future. It's in Vincent's Hospital. But starting, a large research group where you could facilitate, or an institute where you could facilitate interactions between different universities and with researchers from overseas, so we can increase the dynamic scientific environment, and certainly try to make that happen in Australia. That's what I'd love to do. Apart from, of course, doing my own research.
Yeah. What about you, Serena?
Yeah, I will. Yeah, most probably open up my own research institute on an island, on a coral, on a barrier reef. Apart
Nice
from
beaches.
that, so yeah. Exactly. I will probably try to use my funds also to hire younger researchers, and so to include more team members, because as I said, the multidisciplinary is important. So I will try to get more researchers coming from different fields and all working together in the same project, and maybe to include also, I don't know, for instance, on the aspect of the commercialization, to have a regulatory and professional people, even lawyers, working for you and just for you and just for your project, so dedicated to your project, also to speed up the processing and the translation, because as I said, this is a pretty new field for me, but also requires a lot of time, so it requires a lot of people working on a dedicated time for you. So but you need the financial resources to do that, and sometimes paying a lawyer is quite it's quite expensive. So but let's see, my dream
it's much more
institution.
It's much more than just the reagents and the staff itself. It's again, getting all those lawyers and people important for that commercialization or translational aspects is important. And we have seen, with the COVID-19 pandemic, major investment in science can, really speed up and produce some really great results. We've now got a whole host of vaccines available, and that wouldn't have been possible without that sort of investment into making it happen. So as researchers, we're always trying to get the funding we need to do our research. It's competitive, but it just goes to show how important it is. What should we go to next? Go to another general question. There's a lot of talk around, particularly in Australia, that we need more students to pursue STEM subjects and STEM-like careers. From your perspective, what is it actually like to work in research, and what are the benefits and the challenges? Because we do see a lot of this, we need more people in STEM, but maybe fill us in and what it's actually like, and what could be the benefits, and why it would be important. We'll go with Anita.
Well, working in STEM is certainly an exciting area to work in. I'd say one of the benefits too is you learn a wide range of skills. So not only, of course, are you doing the research, but you have to learn things like project management. You have to be a good communicator to be a successful researcher. So you do learn some communication skills. So I think it's not just as simple as working in a laboratory and planning experiments. There's a lot of skills that you attain as well as things like, applying for grant money, managing budgets, teaching. So you really do gain quite a wide set of skills. I think that's one of the advantages of working in STEM. So people will often maybe do a PhD, work in STEM for a while, and might go off to industry and start doing industry projects. And they've really had the benefit of training in a wide number of areas like project management and, of course, laboratory skills in the STEM field. So I think you can apply a lot of the skills you learn in STEM to other areas as well.
Exactly. What about you, Serena?
Another benefit, it can be also the freedom that you have to actually include in your environment many different people and actually understand what language they're speaking. As in my case, I was a simple biologist, and now I'm here doing 3D bioprinting of stem cells and photocross linkable materials. So you suddenly have to, as Anita said, the skills and the expertise that you can acquire are infinite. And I said also the freedom to be always creative and to get the opportunity to actually learn, but also teach to other students, I think is one of the greatest benefits. But in general, research, but especially STEM as well.
I know for I started out in biochemistry and cell biology, and now I've moved into optogenetics, and I'm making LED arrays. I'm doing a bit of electrical engineering. I know how to solder now, all these things. So there's a lot of things to learn, technical as well as, those soft skills as well, that, you're always learning. It's always a new thing to learn. Okay, great. So we've actually got a couple of questions come in again from the audience. We've got one from Gabriella, and they say, hello, I would like to know what kind of physical or chemical stimulus I could use to differentiate mesenchymal stem cells in osteoblasts. So this is for Serena.
Okay, so you're welcome to come into the lab and work with us. So, I can explain. So yeah, we normally use for bone differentiation, we use a chemical stimuli. So through growth factors, hormones. So we basically use cocktails of factors that are present also in the physiological human being, but you can also use a physical stimuli. So, for instance, mechanotransduction is something that we don't actually personally perform in our lab, but is a field that it has been investigated quite a lot. So, to actually perform some mechanical stimulation onto the cells, and especially the mesenchymal stem cells, they can react really well through specific receptors that they have on the cell membrane, and to actually differentiate thanks to this physical stimuli. There are also electrical stimulation that you can perform. As I said, we attempted to do that with our adipose-derived stem cells. It turned out that the adipose tissue is not the best source for the cells to become actually electrically stimulated towards bone, but other niches like the bone marrow niche is one of the preferable ones to actually convert both chemically and physically the stem cells into bone-like, I would say bone-like mature cells.
Okay, great. And we have a question from John. How important is it to increase investment in innovation and technology to prevent our best scientists from going overseas? I think he's touching on a lot of researchers, for a period of time will usually go from Australia, will go overseas for postdocs and training, that sort of thing. So Anita, what were your thoughts on this?
Oh, I think it's extremely important. And as like Serena was saying before, I think we need more money to bridge that gap in between science and translation. There is a push towards that at the moment, but one of the things that's very important is not to forget about the basic science and the science behind that too. So I think, a lot of researchers do leave to go overseas because sometimes in certain countries there's a bit better support of that, but I think it's becoming better in Australia. But yeah, it's, of course, increased funding is very important. No doubt.
Yeah, what about you, Serena?
Yeah, so I'm actually one of the brain escaping from my country. So I actually went overseas. So I'm actually overseas, but it's not necessarily a bad thing. So actually, I can always go back to my home country and restart my own research in there. So, but again, pending funding, of course. But I can still collaborate with my home research lab, so there are opportunities to actually still exchange thoughts and technical skills and projects and also a little bit of money in between countries. So it's not necessarily a bad thing to go overseas even for a while or forever for good. I don't know. But I see positive things in getting away from your country and explore other different cultures and a different type of doing science.
Yeah, exactly. And we're very lucky in that science is quite a global effort, and there are lots of opportunities. I guess it also depends on the person as well and their area of research and what they want to do. But yeah, obviously we're more investment in being able to provide good opportunities for young researchers in Australia, I think is important. I also want to ask, oh sorry, you go.
Yeah, I was just going to say, I think the funding's important across the board. I think, like you were saying before, funding young research is very important, but also funding keeping older scientists that can mentor these younger scientists and have a lot of the knowledge, I think, is very important too. They can't be, not saying they're forgotten, but that needs to be facilitated as well.
Yeah, exactly. Sustainment at all those levels is important. I'll move on to a question. What inspirational message or advice would you give to young people pursuing STEM or biomedicine? What do you think, Serena? What would you say?
Yeah, well, of course, one message is that if you want to take this career, of course, resilience and persistence and perseverance are one of the skills required, together with passion and also commitment. But yeah, keeping in mind that there are great benefits out of this STEM career or academic career, and also more commercially or translational career in science. So as Anita said at the start, so there are great opportunities because you ended up maybe starting with doing a PhD or a postdoc, and then you work for a company making medical devices. So you never know what can happen into the future, but of course, resilience and this sense of never giving up on things, because you never know when the most difficult thing will happen. So I remember that my professor at the high school, math professor, used to say, what is the most difficult exam is not the current one, but it's always the next one. So I think this is important to bear it in mind. And but there are great benefits in pursuing this career. So despite this, it might be challenging for financial point of view and personal life point of view, but there are great benefits, and I'm still convinced about my choice, and so good things will happen.
Seems you're still very hooked on science.
Yes,
which is good.
I am.
And what about you, Anita?
I totally agree with Serena. I think she's spot on there. I think as well what she said before too about nothing's really ever a failure. So, if you feel that your experiments are failing or things are not going the way that you expect them to, you have to keep an open mind. It's actually telling you something. So everything's a discovery. Even when things fail, it's telling you something. And it comes back to what Serena was saying before about resilience and being open, in my opinion, being open-minded too about what you're seeing. You have to keep an open mind. Yeah, but I certainly agree with Serena too.
Yeah, and just to extend on that, I know it's not something necessarily everyone's instilled with, that strong resilience. It's something you do develop over time, and it's just something you have to be patient with, but really work out to, it can give a lot of reward. I know I've certainly experienced that. It's just something you do develop over time. We have another question from the audience, from John. How important is it for all of us to become more scientific? So I'm guessing, more to think like a scientist. What do you think, Serena?
Yeah, I think it's equally important to think about also an art mind. So a creative mind is equally important. So not necessarily always scientific. So as Anita says, so it's important to develop other skills beyond the scientific mind or the mathematical and modeling physical mind. So in this work, you have to learn how to actually present your data, write a report, write an article explaining your discoveries, write grants applications, so to convince people that your research deserves to get granted and to get financed. So I think it's equally important to have an open mind and to actually develop other skills, so not only the scientific mind.
Exactly, that creativity is very important.
Yeah.
And for you, Anita, what do you think?
I agree. I also think, especially these days, bearing in mind, I didn't want to mention it, but the pandemic, I think it's very important to keep, in terms of a scientific mind, the way scientists operate is they're very much evidence-based. So nothing's hearsay. And you've always got to look at reputable sources for information. Have they done their research properly? I think, to me, it's more and more important that people be scientifically minded, and I actually think we should teach, things should be, a little biased, I guess, but things should be a little bit more scientifically orientated in schools as well. I think, yeah, so I do think it's very important to have a scientific mind, especially these days, in being very careful about where you get your information from and making sure it's from reputable sources.
Exactly. That misinformation, got to try and avoid that as much as possible.
Yeah.
Especially what we've learnt from the pandemic. Okay, fantastic. I think we will wrap things up. I want to say a big thank you to both of our speakers, Anita and Serena. It was really great to be able to learn about your research and also talk, career and opportunities as well. We really appreciate it. So I'll move on to thanking our sponsors who have generously supported us and make CSN and these other events possible for us. And for the audience, I'd like to just let you know, if you are able to please take some time for a 2-minute survey, which will be sent around. Your feedback is really important to us, and it really makes sure that we can continue to deliver really worthwhile and informative events to the community. There'll be a guide after the live stream to complete that survey. And finally, I'd like to ask attendees to consider subscribing to the CSN YouTube channel by clicking on the subscribe button, which will also appear below. So thank you, everyone. We hope you have a wonderful day. And thank you again, Serena and Anita, for a great discussion.
Thank you, Christina.
Thank you
So much.